Hello and welcome everybody to another exciting episode of SciTalks 2.0. As always, my name is Dr. Jack Saxenian, and today I have an exciting guest for us to talk about a possible breakthrough drug for the treatment of COVID-19, SARS-2 COVID virus. Before we get into that, I just want to quickly give an update about some of the recent updates on the research front for the vaccine development. Uh, Oxford University and AstraZeneca, they had paused the vaccine trial for the reasons that were mentioned in a previous post that I made on Facebook, but essentially they had an adverse event in one patient, so they stopped the trial, but they've announced that they've since resumed the trial, which is a good sign that the vaccine is going forward with that. And in the meantime, outside of the vaccine, there's been a lot of push to come up with therapeutics to help treat and overcome some of these issues related with the SARS-CoV-2 infection. So with that, today I want to talk about a specific drug that is very important, and I say that half jokingly because the name of the drug is actually VIP, which is vasoactive intestinal peptide. And I have with me a guest who is foremost expert on it, has spent over 20-something years studying this, and I hope I'm not dating you too much, Kato, but it just to tell the people and the viewers and the audience about your level of expertise on this specific peptide. So we will be discussing about this peptide, where it is in the clinical trial stage, and for some of the viewers that are more scientifically and medically inclined, we'll go into the mechanistic details of how this peptide works. So with that, I will quickly introduce my guest and then have her give a quick background about herself. So Dr. Catalina Abad is a scientist who has a long history of research on this VIP drug and has published numerous papers, groundbreaking work on that. And she's a trained immunologist, so she knows every aspect of this. She'll go a little bit into what VIP is, and I'll add a little bit more about the clinical trial stage of the drug at this point. So without further ado, please, Dr. Abad, kind of introduce yourself, and then we could continue talking about this possible new drug for SARS-CoV-2.
Okay, thank you. So yeah, you're right. I've been working for more than 20 years now on VIP, and it's very related peptide PACAP. I did my PhD in Spain, and the objective or the goal of my PhD was to demonstrate that VIP and PACAP have immunoregulatory properties. So actually, VIP and PACAP are neuropeptides. So these are substances produced by the nervous system, and their actions in the nervous system were very well known for many years. And my lab demonstrated, actually, it was one of the first ones to demonstrate that these neuropeptides could have actions on the immune system. And one of the most important actions is the inhibition of inflammation. They are anti-inflammatory peptides. So during my PhD there in Spain, we tested the effects of VIP and PACAP in chronic inflammatory diseases, rheumatoid arthritis, and Crohn's disease, which is a chronic inflammatory disease of the joints for arthritis and of the intestine for Crohn's disease. So then after I moved on and I did a postdoc at UCLA for quite a few years. I started as a Fulbright postdoc for a year, and then I was assistant researcher working with mice that do not have VIP and PACAP. I forgot to say it, but VIP and PACAP are naturally produced by the body since they are neuropeptides. So we had mice that not produce them. And my goal was to study what happened to these mice. Did they lose the protection by VIP and PACAP against inflammatory diseases? And at UCLA, in Jim's Warheck's lab, I studied the response of these mice to models of Crohn's disease, of septic shock, which is an acute inflammatory disease, and the very well-known model of multiple sclerosis, which is EAE, or experimental autoimmune encephalomyelitis. And we found that when you lose VAP or PACAP, you have hyper responses, hyper exacerbated disease. And then after that, I moved to France with a position as a research engineer, where I keep working on autoimmune diseases, not only VAP and PACAP, but other aspects of autoimmunity.
All right, Kat. So that's a great summary, which I'm sure is cut into 1/10 of exactly all the work you've done. So can you tell the viewers why we've been excited about it and why I've reached out to you besides wanting to talk to you again as friends and colleagues about VIP in the context of the ongoing pandemic, why there's some excitement going on?
Because during a COVID infection, so there is a first phase where the most important is the viral infection, the effects on the pulmonary destruction, and there is also an ongoing immune response. And at some point of the disease, the patients get very sick, but not only because of the virus infection, but because the immune system, the own immune system, hyper responses to the viral infection is what is called a cytokine storm. So the immune cells produce too many cytokines, it is too much inflammation, and this is what actually has a very negative effect on the patient. They basically kill the patient. So we need to, at this point of the disease, we need to provide molecules that can inhibit inflammation, anti-inflammatory. So that's why VAP came into mind of some doctors as a potential interesting molecule to decrease the cytokine storm.
And to add to that, so far it's been tested in, I believe, 5 patients already, and in really sick stages of the disease in these 5 patients. The first patient was treated at, I believe, the Houston Methodist down in Texas, and they showed that patient recovered really well. They also looked at their oxygen saturation and their ability to breathe normally, and all of those were reversed. So since then, there's been clinical trials that have started, and the FDA gave a fast track IND for its use to be tested in a clinical trial, and also allowed it to be used under emergency use. So people are using it in these very sick patients. The clinical trials are ongoing. There's multiple institutes involved, and the results of the studies will come out, I believe, end of September, early October. But from the initial preliminary data, it looks like it's very good, at least in a very small sample of people. And as we were discussing earlier, we shouldn't get our hopes up a lot, but based on mechanistically of how VIP works, and we'll go into it a little bit more detail, we think that it has very good potential, right?
Yeah, so the advantage of VIP is that it acts on many different pathways, inflammatory pathways at the same time. So there are drugs like the anti-TNF alpha, the infliximab, and other, which is an antibody against TNF alpha, that block one certain cytokine, for example. The advantage of VIP is that it blocks, for example, one main inflammatory molecular pathway, which is NF-kappa B, and by inhibiting NF-kappa B, it blocks several cytokines at the same time. In the same time, we had seen in mice and in vitro that VAP can promote anti-inflammatory cytokines. So it doesn't only block inflammation by blocking the pro-inflammatory cytokines, but it promotes anti-inflammatory cytokines like IL-10. So it's basically a very complete molecule that acts on many different pathways at the same time. So that's the advantage against other drugs.
And so when you say pro-inflammatory and anti-inflammatory, right? It's not like we need one versus the other. It has to be sort of like a stepwise controlled immune regulation, right? And we think that in the context of COVID-19, you have the initial phase of inflammation, which you need to fight the virus,
Mm-hmm.
and
That's
then
what I
it
said.
gets out of control.
You need to block inflammation in the later, in the COVID infection, in the later steps. In the beginning, you need, that's why there is inflammation. You need the immune
Right.
system to activate it to finish up with the virus. But the problem is that at some point it gets out of control, and that's when you want to decrease it. Yeah.
So, in the context of COVID, we don't have any drug outside of steroids in a very specific population of people that actually decreases mortality, right? Because most of the drugs tend to act on potentially one pathway versus the other, and the timing is very important. So, I think this is why it was exciting that VIP in very late stages of the disease in very sick patients could potentially have a benefit. But having said that, there's also some studies showing that it has other effects, right, in terms of viral replication and protection to lung injury in certain models. So can you touch upon that a little bit?
Yeah, actually, this, well, I work most of my life on the inflammation, anti-inflammatory effects of VIP and PACAP. And yes, I saw recently when I heard about these VIP COVID effects, I read that it can have apparently some antiviral, anti-replication effects. And it took me to some previous studies on HIV where researchers have shown that actually VIP was able to decrease HIV replication in peripheral blood mononuclear cells, so in human cells in vitro. So it seems that it could have this double effect, antiviral and anti-inflammatory, so it has a lot of potential in the disease.
So in that context, then I think it could be good. Do you think in terms of the early stages of the disease as a potential antiviral effect, and then for the second stages of the disease, anti-inflammatory effect?
This will have to be seen, I guess. Timing is very important, I guess, because you need inflammation in the beginning anyway. So you wouldn't, in my opinion, I'm not I'm a researcher, so I'm not a clinician. I say, in my opinion, I think the right moment has to be found. Maybe not too early because you need some inflammation to be ongoing, but not necessarily to the very sick patients. But it's something to be tested, of course, in the clinical trials.
So I know you've done some work on sepsis because this is also one of the things that can kill patients in COVID. And also, can you touch a little bit upon the rheumatoid arthritis model? Because I believe like these are very closely related to what we think is going on in COVID-19 patients, specifically with ARDS, which we think might or might not be happening. But in terms of the cytokine milieu, let's just say this is what is going on. And how are these models potentially similar? And therefore, what you've done in terms of your work on the previous models could be translated to what's going on now.
Yeah, so I guess in this case, it's closer to the septic shock model than arthritis, because arthritis is a chronic inflammatory disease where there is a big component by autoimmune T cells. I think in COVID disease, we're talking more about acute inflammation, if I'm not mistaken. So this is more what happens with a septic shock or endotoxemia. And yes, in mice, we saw that even a single dose of VIP was able to increase the mortality, the survival, decrease the mortality of the mice that had been treated with LPS, which is the model we use to induce septic shock.
And in that septic shock model, what did you see it do to the specifically to the immunomodulation in terms of specific cytokines or specific T cell populations? Did you look specifically into those?
So in the septic shock model, we found that VIP decreased the levels of TNF alpha, of IL-6, IL-1, which are the main pro-inflammatory cytokines. And then in the model of arthritis, we have seen other cytokines that are decreased, like IL-18, IL-12. Basically, I would say that every cytokine that we looked at, pro-inflammatory, was decreased by VIP. Also chemokines. So chemokines, as you know, they attract immune cells. They are used to attract immune cells to amplify the immune response to the site of inflammation. So all cytokines that we looked at were decreased by VAP. So VAP decreases the ongoing inflammatory response by decreasing cytokines and also by blocking the arrival of more immune cells that are going to maintain the inflammatory response. So that was about acute inflammation. In chronic inflammation, we also saw that VAP modifies the response of the autoimmune T cells, which are maintained for a long time, which are switched from Th1, which is inflammatory type of T cells, to Th2, which are anti-inflammatory. So VAP was capable to modifying the response of the autoimmune T cells from pro-inflammatory to anti-inflammatory, from Th1 to Th2.
So two quick questions in regards to that. Did you see any effects on Th17s? Because it seems like they're playing an important role in the context of COVID-19. There seems to be misregulation of Th17. And also, I just recently read a paper that profiling neutrophils and studying the neutrophil signature could tell which patients are going to do poorly versus ones that are going to do better, relatively speaking. So it seems like neutrophils play a very significant role in the disease pathophysiology of COVID-19. So did you see any effects of VIP on these cell populations?
So for Th17, we didn't study directly in vitro the effect of VAP on Th17 cells, but in the models of disease that are used in arthritis, multiple sclerosis, we saw that VAP decreased the Th17 pathogenic response, because as you know, Th17 cells can be pathogenic or non-pathogenic depending on the cytokine expression. So not directly, but indirectly, we have seen an effect, a decrease in the Th17 pathogenic response. And about neutrophils, actually, nobody has really studied VAP and neutrophils. Well, I would say nobody. There are some old studies, controversial, with both beneficial and beneficial, positive and negative actions. So I don't think there's much done about that, I mean, to my knowledge.
So there's still a lot that needs to be investigated. But in terms of the overall big picture, if I understand you correctly, that the VIP pathway could, I mean, especially IL-6, right? That seems to be very important. Although so far, anti-IL-6 antibodies that are being used in COVID patients haven't really panned out to be that great in the clinical trial stage. But that could be because we're only targeting one cytokine, right?
Exactly. This is what I was saying, that when you act with VIP, you act on many different cytokines at the same time. So that's the advantage versus other like focused therapies.
And so why is it that in the body, if the people are not normally expressing VIP, why would it get out of control then in these patients? I mean, I know this is a loaded question and you may not have the full answer to it, but do you think that there's a lack of signaling going on, or there's dampening of the signal, or what's potentially going on? Maybe you're septum.
Yeah, I just think that the levels of VAP that are produced are not enough to cope with the inflammation that is ongoing. I think I read, I'm almost sure, I read that the COVID patients have increased levels of VAP when they're very sick. So like the body is trying to produce VAP to decrease inflammation, but the one that produces is not enough. So I think that's the reason why
It's a level effect. I see.
Yeah, it's a level.
And the other interesting thing is, like, there's these studies actually showing that there may actually be an autoimmune component in COVID, because we're seeing certain autoimmune kind of diseases popping up in COVID patients. So there might be an actual autoimmune component to it, not in all patients, but in some patients, right? We're seeing, I believe, autoimmune related to the central nervous system. We're seeing some autoimmune diseases related to that. So if VIP could alter that as well, it might be a good, maybe prophylactic, something that could be treated for patients who have the disease. So overall, based on the studies that you've done, they're mostly been on mice and in mouse models. So do you know of any studies in human, either tissue or in cell cultures, or even in human patients, that you could potentially help add more weight to why we're using this in COVID-19?
I know the team of Rosa Gomariz in Spain, who is my PhD mentor. So they keep working on VIP and arthritis, but they're basically they're focused on using the levels of VIP as a biomarker of the disease. They measure the levels of VIP of patients at different stages, and they have seen that actually there is a relationship between low VIP levels and a worse prognosis to the disease. Um, this is the team that I know that they're doing most of the studies in human. I think the team of Mario Delgado, they have also been involved in sarcoidosis. There was a clinical trial in, I think it was phase 2. And they had, well, you know, sarcoidosis is a lung disease where there's inflammation, and they had some positive results. I don't know where they are in this clinical trial, but directly inflammation in the lung, so it could have really to do with the COVID infection, and that's all that I know.
Yeah, and so I think overall, if I get the sense that the amount of information that we know about this drug and or the peptide and its signaling pathway is definitely good enough for a clinical trial to go forward, right? And add this to our armament of potential medications that we could give to COVID-19 patients. So what are some of the, let's just play the devil's advocate for a minute. What are some of the negative things that you anticipate could happen from treating patients with COVID-19 in terms of altering their immune response improperly or wrong stage? And things that people should maybe be worried of. I mean,
In
physicians.
terms of the immune system, when we used it in mice models, we never saw any secondary effect. We were questioned many times about possibility of getting infections because you block the immune system. You can't do infections. We have tested it, but we did not see any abnormality of the microbiome that was treated with VIP. As like we were talking before, in general, VIP is known to be vasodilatatory, and people having concern about inducing hypotension or tachycardia, but we did not observe this in mice except with very high doses of VIP, was something that could be considered. No, I guess only question is because VIP inhibits this Th1 pro-inflammatory and may direct towards Th2. Is it allergy related? Could it lead to allergy, eczema?
But in an acute setting, I don't foresee that as being a big.
No, in an acute setting, I don't think so. It has to be seen in humans because sometimes from mice to human there is unexpected differences. But no, I don't foresee any major effects.
I see. Okay.
And also, hold on, I think this VIP now drug that they're using, Aliptadil, is using against for erectile dysfunction, and I don't think there has been reported any major secondary effects for the drug.
So far, I haven't read any adverse events or anything specific in any of their use. And I know we were discussing a little bit earlier that there is some studies of VIP in the context of septic shock in humans, not necessarily COVID-19, but in humans.
Yeah.
Can we just touch upon that really briefly, just so that people have an idea of what has been done in the human context?
There was one clinical trial, but it was in 1998, so it's pretty old. That was done for septic shock at the Stony Brook Institute in New York. But the number of patients that enrolled was pretty small, and it was patients in very advanced stages of the
Mhm.
disease. And also, I think I heard that there was no placebo control, so it was a study. They didn't show any effect of VIP. There were no negative effects, but no effect. But at the same time, the patients were in such an advanced stage that,
Mhm.
well, it's difficult to have a benefit when they're already
Mhm.
so far along.
Um, all right, awesome. So do you have any other last words or comments or things that I may have forgotten to ask you that you want to additionally add to people who are watching this, either the lay people or the physicians, scientists that want to know a little bit more mechanistically about how VIP actually works?
No, I just want to say that I'm very happy that there's some interest on VIP, because for many years we have been demonstrating its many labs, of course, me also, but all labs have demonstrated its beneficial effects in so many inflammatory diseases. Like I said, sclerosis disease, septic shock, multiple sclerosis, but also in recently thyroid disease. There are many different diseases. So I'm just really hoping that this is going to wake some interest of people for the use of VAP on many other diseases. And PACAP, it's related, the peptide also has a lot of interest in, has been tested in neurological conditions in mice, like for Parkinson's disease or Alzheimer's, mostly because it's neuroprotective, but PACAP is also anti-inflammatory. So PACAP could also have could be very promising for these diseases. So I would like people to be aware of the potential of these
Mhm.
peptides.
And just really quickly,
Yeah.
I know we talked a lot about what it's doing, how it's doing it. Can you just describe to the viewers how it actually works in terms of its signaling, what it does, just in case people are interested of like, what is it, like how it actually gets the information from the outside of the cell to the inside?
So VIP has three receptors, which are VPAC1, VPAC2, and PAC1. It's believed that its immunoregulatory properties are mainly through VPAC1 and VPAC2, whereas PACAP is involved in other actions. So basically, VIP binds to its receptor and is going to induce the production of cyclic AMP, which is a signaling molecule that in turn is going to lead to NF inhibition. So that's the way. I
So,
don't know
so,
if you
uh,
know.
so at the end of the day, all the signaling comes down to NF kappa B, and we know NF kappa B is great for
Mm.
immunoregulation. But it just seems like NF kappa B is also such a vastly active signaling pathway that how do you get the specificity from the VIP signaling?
Because the VAP and PCAP receptor is not, or VAP receptor is not expressed in all cells in all times. For example, VAP receptor is expressed in immune cells, for example, T cells express VPAC1 constitutively, and VPAC2 is not expressed, but when they get activated, it gets
Mhm.
upregulated. So all these interplay between receptors expressed not in all immune cells, but in certain immune cells, depending on the activated status, is going to lead to the fact that it acts on specific immune cells that express the receptor at the moment, at the right moment.
So the fine tuning is done by the level and the timing of expression of the receptor, whereas the downstream is more of a master regulator like NF
Mm.
kappa B, and that switches the immune response drastically from one direction to the other, and so in that context, then it seems like even one of the criticism that you were talking about that people had been saying is that because it's a peptide, it gets used, broken down, and um, essentially excreted pretty quickly by the body. But because of this signaling pathway, it seems like its effects could linger around for long periods of time, even if the peptide isn't around anymore.
Yeah, of course, once the peptide binds to the receptor, the signaling molecules are released, and then you have an ongoing effect for like long lasting.
So then in terms of drug delivery, we should expect it not to be an issue for, long term. It's not like you need to be constantly pumped with this drug for long periods. Although the clinical trials have like 3 different dosing that they're doing, and escalated dosing. But I believe it's just one-time injections. I don't think they're continuous
It's not like a pump.
injections that they're doing. But even with, I believe just injections, they got pretty good results. So it seems that it has a profound effect on the immune system. Because I'm still not convinced about it affecting the viral replication aspect of it at this point. Because you know I just feel like at that stage of the disease, the viral replication, I don't know how much of a role it plays. But definitely the immunomodulatory and the protection of the lungs and the type 2 alveolar cells, which are the cells that the virus infects. And interestingly, these are the cells that express the receptors for VIP, right?
Yeah, exactly.
And they provide the protection. All right, Kata. So this has been great. Kind of a really good summary of VIP and its actions, and some of the studies and the background on that. So hopefully the viewers will have learned a little bit. At least I know I keep learning more and more each day. So I want to thank you for that. And of course, if anybody has questions, they could drop a comment below, and I'll do my best to answer. And if not, I'll reach out to Dr. Abad for further answers. So with that, Kata, I want to thank you again, and I want to thank you for doing the good
Thank you.
work and the groundwork for potentially coming up with a drug that we never anticipated, the pandemic. But this is how science is, right? You do
I
the
know.
work, and one day you'll see the
But
effects.
that's great. That's great.
Well, thank you so much, Kata, and
Thank you.
good luck. Stay safe and be healthy.
You too.
Bye.
Thank you.